Okay, before we begin, are there any questions? Not hearing anything, so I'm assuming there are no questions. So let's begin. First, can somebody confirm you can hear me?
Yes.
Yes.
My voice is a little raspy, so I'm probably not going to be as loud, and I apologize for that. If you need me to repeat something, please just ask. I don't know if this turns up my microphone or not, but let me see if I can turn up the microphone. I don't see how to turn that up, so I'm not going to try and figure it out now. All right, let me share my screen. I believe we were here on chapter 8. Today is February 22nd, and we're supposed to be on chapter 8, and we're behind schedule. We should be about 2/3 of the way through chapter 8, and we're actually only, well, less than a third of the way through chapter 8. So I'm going to try and move quickly today to try and make up a little bit of time. Like I said, if you want something repeated, just ask. Let me remind you, your infectious disease project is due this Saturday at 11:59 p.m. There is a lab today for those who are coming to the 7 p.m. lab. All right, let's go on with the lesson. So we had talked about DNA. And how it's normally two strands. The two strands are held together by hydrogen bonds. A single strand of DNA is held together by chemically covalent bonds. When we're talking about DNA, you can talk about the flow of information within a cell, and that is usually called the vertical flow of information. A DNA contains the information, and this is known as the central dogma of biology. DNA codes for the information. That information is changed by transcription into the information coding in RNA. And most of the time, that information is translated into protein, and then the message or what's coded in the DNA will be coded in the protein. DNA, RNA, and proteins each have their own language, and they each have their own way of storing the information. When we're talking about the flow of information, you can talk about the flow of information within a cell, which I just talked about, but you can talk about the flow of information between cells or between generations, and that would be like from parent to offspring, or from parent cell to two daughter cells. The flow of information from, let me blow this slide up. The flow of information between generations of cells has the DNA coding the information, and then that DNA is replicated, and then one chromosome of DNA moves to one side of the cell, the other chromosome moves to the other side of the cell, and then the cell splits, each daughter cell getting a copy of the original information from the parent cell. The information in DNA can also be said to flow between cells of the same generation. And that means the DNA from one cell, the recipient cell, loses that DNA, and the DNA can go out in the environment, and then move into a new cell that we call the recombinant cell. And then the recombinant cell can express the DNA of the recipient cell. So cells can gain DNA from another cell, and that is called the flow of genetic information between cells of the same generation. Any question about that? You'll note that I changed the way the author talks about the flow of information between cells of the same generation. And that is I crossed out the line here because oftentimes this parent cell dies and then the DNA comes out of that cell and it's free DNA in the environment, and that DNA can be taken up by the recombinant cell. It is possible that this cell can give a plasmid directly into that cell, and we'll talk about that much later in this lesson. I'm not sure why they call that recombination, but maybe because this is the recombinant cell, but this is not normal DNA recombination. It's taking up DNA into the recombinant cell. Anyways, this tells you that the flow of DNA, the information in DNA, can happen in 3 different ways: within a cell, between generations of cells, and for humans, we'd say from parents to the offspring. And it can also flow between cells of the same generation, where the DNA can leave the donor cell and then be taken up by the recombinant cell. The flow of information within a cell happens by the central dogma, where DNA has the information, flows to RNA, which flows to protein. The language of DNA is DNA nucleotides. There are only 4 letters in this alphabet. The language of RNA is RNA nucleotides, and so it's a very similar language to the DNA. And once again, there are 4 letters in this alphabet. For the language of proteins, the language is in amino acids, and there are 20 different letters in this alphabet, meaning there are 20 different amino acids, at least in the human species. For comparison, English has 26 letters in its alphabet. Any question about any of that? So looking at this once again, we can say that the information in the DNA is, that is a dog, the original information coded by the DNA. In transcription, it would be something similar, but a little different accent. That is a dog. And then in protein, after translation, the information would be something like quello è un cane, a totally different language, but containing the same information. Any question about any of that? All right, so that's it for the flow of information within a cell. Let's talk about the flow of information between generations of cells. This is often called the vertical flow of information because the information is flowing from parent or parent cell to children or daughter cells. And this one is called the horizontal flow of information. Any question about any of that? Hopefully you didn't see any of that. The flow of generation between generations of cells, or from parent to offspring, follows the replication of DNA, and DNA is replicated semi-conservatively, where the 2 strands of DNA split into a single strand, and then the RNA polymerase binds to the strand of DNA that it uses as a template, sees a T there, and then asserts inserts an A, the complementary base pair, to the T on the newly replicated strand of DNA. And then the DNA polymerase moves down to the next nucleotide, which is a G, and it will insert a C. And then it moves down to the next nucleotide, sees a C here, puts the correct complementary base on the new strand of DNA, which in this case would be a G. We call this semi-conservative replication of DNA, where the two strands split and then one strand, each strand, but one strand is used as a template to replicate the DNA on the newly growing strand of DNA. So DNA replication is semi-conservative. That means that if the original strand of DNA were two black strands, the new strand of DNA will have one parent black strand and then one newly replicated strand of DNA. And then both will be like that. And that's semi-conservative replication of DNA. That's different from the conservative model of DNA replication, where in the two daughter cells, one daughter cell would have the two parent strands of DNA, and then the other daughter cell would have two strands of newly replicated DNA. And the conservative model of DNA replication doesn't happen. DNA replicates with the semi-conservative model. And don't ask me what the liberal model of DNA replication is, because I don't know. How DNA is inserted is that the DNA polymerase binds to the A on the template strand, which is the parent strand of DNA, and it has to be a single strand for the polymerase to bind to it. And then it inserts the correct base pair and adds it to this growing strand. And how this strand becomes longer is it requires energy, meaning whenever you make a bigger molecule, you have to add energy. And the energy comes from splitting off two of the phosphates from the nucleotide that's being added. So this is, what would that be? TTP, thymine triphosphate. And two of the phosphates are broken off, meaning that, come on, mouse, this bond here breaks off, and then this nucleotide, the T, the phosphate, and the ribose are bound, and that's a chemical bond. The phosphate binds chemically to this OH group on the ribose, so that's a chemically covalent chemical bond. Any question about that? Remember, the DNA being held together happens at the nitrogen-containing bases, and the two strands are only held together by hydrogen bonds. We'll talk a little bit more about that. And so the thymine, come on, mouse, binds, meaning it binds double-strandedly to the not double-stranded, it binds with a chemically covalent bond to the single strand of DNA, and then to the double strand of DNA, it only binds with hydrogen bonds. And then the phosphates come off, and when they come off, they release energy, and that's what allows this reaction to proceed. All right, let's watch a little video on DNA replication, assuming I can get it to load. Let me end this show here and see if that's loading. Yes, it is.
DNA is a molecule made up of two strands. twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, any questions about that movie? It did cover a lot of information altogether. We'll go over it. But any questions? All right, I'll leave that there just in case I want to get back to anything. Not sure why we're there. We should have been here. So let me blow this slide up. So DNA replication only happens on a single strand of DNA. And normally in our cells, the DNA is double-stranded. It can be single-stranded, but it only is single-stranded during DNA replication and also during RNA transcription. So how the DNA becomes single-stranded is we have a helicase bind to the DNA and then open it up from being double-stranded to being single-stranded. The DNA polymerase will then bind to the single strand of DNA and begin copying it using the single strand as the template to build the new strand of DNA. Any questions about any of that? So the first protein that we mentioned was helicase. The second protein or enzyme we require in this process is the DNA polymerase. There is a third enzyme I do want you to know, and that's the enzyme ligase. And ligase sort of finishes the process, and we'll talk about it in a little bit. The other enzymes, I do not care if you know them or not, like primase, uh, the making of the primer and then it's RNA. I don't care if you know about it because the primer has to be removed, but it does start, meaning that when you first start replicating DNA, you do need to put in an RNA primer to get the DNA replicated. And then the DNA replication proceeds, and then that RNA primer is removed and replaced with DNA, which is a fairly complex way of doing it, but that's the way the cells do it. Let's see. So one strand of DNA can be made continuously, and that is the leading strand. And that's because the DNA polymerase moves down this strand in the 5 to 3' direction. And so the DNA polymerase can just continuously move down that strand, adding nucleotides to it, because this strand, the leading strand, is in the right orientation. The other strand is in the wrong orientation, and to replicate it, the strand is separated, the DNA polymerase then binds to it. Let's put it here. And instead of moving this way down the strand the way the helicase is moving, the DNA replicase moves in the 5 to 3' direction, going backwards, replicating the DNA. And that results in Okazaki fragments because the DNA is replicated in short fragments backwards, meaning we have one fragment here, one fragment here, one fragment here, one fragment here, and then there'll be another fragment right here. Okay. And the RNA polymerase binds here and then moves this way. And then once it reaches where the other fragment of DNA is, the DNA polymerase stops and falls off and then combine to the DNA again to replicate the DNA. Now, these Okazaki fragments are not linked to each other by the DNA polymerase. So when it starts this way, I guess I should clarify that. When it starts here with the first nucleotide it's adding, and we're just skipping the RNA primer for the time being, and then the DNA moves to the next nucleotide, that is linked chemically by a chemically covalent bond. And as long as that DNA polymerase is moving, adding nucleotides, the nucleotides are linked together by a chemically covalent bond. But when it comes to the last base, which I guess would be right there, it links this way, but it doesn't go forward, linking this Okazaki fragment to the newly replicated Okazaki fragment. And I think this could be being synthesized when this one is, although usually this one finishes first because this one got started earlier. And so wherever you have an Okazaki fragment, you have a gap in the single strand of DNA to the next Okazaki fragment. And so I guess what you could say is each Okazaki fragment has a gap at the place where it starts and at the place where it ends. And that gap will be filled in by ligase. It will bind to wherever there is a chemical bond that has not been made between the DNA, like the DNA of this Okazaki fragment and the DNA of that Okazaki fragment, and fill in that chemical covalent bond. It does not add a nucleotide. All it does is it combines this nucleotide in a covalent bond. Come on, mouse. Combines this nucleotide to this nucleotide here. So combining this Okazaki fragment to that Okazaki fragment. And the DNA ligase works a lot on the lagging strand. On the leading strand, it may only work one time. And it depends if the DNA polymerase, if it's beginning at the very start of the DNA, actually, if it starts at the very start of the DNA, and then it stays on the DNA strand until the DNA ends, there will be no cases where the ligase works. However, the DNA polymerase can move along here and then fall off, and then another DNA polymerase can bind here or someplace near here, and then move forward. And where the first DNA polymerase strand was, and then the second strand, that will not be linked together. And the ligase will link those two strands of DNA together, the same as it does the Okazaki fragments. So the DNA ligase does not work very often, if at all, on the leading strand, but it works, well, twice for each Okazaki fragment on the lagging strand. Any questions about any of that? And the reason for the Okazaki fragments is DNA polymerase adds the nucleotides in the 5' to 3' direction. So it can just add it continuously on the leading strand. But on the lagging strand, the DNA polymerase has to move backwards. And so there will be many short Okazaki fragments. And Okazaki is named after Dr. Okazaki. I could be mispronouncing that. I'm not Japanese. And I don't know how to speak Japanese. And he's the scientist who discovered it. Any questions about DNA replication? All right, I talked about the leading strand where it's synthesized continuously. The lagging strand is synthesized discontinuously, resulting in many Okazaki fragments. When the DNA is put in and has RNA primers in it, the RNA primers have to be removed. And they will be removed by DNA polymerase, and a DNA base will be put in as the RNA polymerase, the RNA base is removed. And then the Okazaki fragments, as well as any discontinuation in the leading strand, will be ligated together with DNA ligase. So the three important enzymes: DNA ligase, DNA polymerase, and helicase. Any questions about any of that? All right, let's move on to RNA transcription. RNA transcription is the synthesis of RNA from DNA. Unlike DNA replication, where all of the DNA is copied, only the genes of the DNA are transcribed. Everyone get that? So not all of the DNA is transcribed, only the genes are transcribed. Now on a bacterial cell, most of the DNA codes for something, so most of the DNA is genes. Very little of the DNA is non-coding DNA. However, on a eukaryote chromosome, most of the DNA is non-coding DNA. It does not code for a product, a gene product, which would be either a protein or an RNA. A DNA is transcribed to make RNA, and there are three types. There's messenger RNA or mRNA, transfer RNA or tRNA, and then rRNA or ribosomal RNA. I'm trying to remove that. Oh, there, I finally got my mouse. There we go. Transcription begins when the RNA polymerase binds to the promoter of DNA. We'll talk about promoters later. Is it in this lesson? I guess it is later in this lesson. And that is, a promoter is a region of DNA that codes for the RNA polymerase to bind to it. Transcription then proceeds in the 5' to 3' direction, meaning that RNA is made in the 5' to 3' direction. Transcription stops when the RNA polymerase reaches the terminator. The terminator is a region of DNA after a gene that codes for the RNA polymerase to fall off. So the promoter codes for the RNA polymerase to bind to it. The terminator codes for the RNA polymerase to fall off. The promoter is before the gene. The terminator is after the gene. And usually the terminator is immediately after the gene. I'm not sure if it's always immediately after the gene, but usually, at the very least, it usually is right after the gene, immediately after the gene. The promoter is usually upstream of the gene, meaning it's not immediately upstream of the gene. It's upstream a ways, I don't know, several to many nucleotides above the gene. You don't need to know that, but just giving you the information. This is showing you RNA transcription with RNA polymerase, and there is a, come on, mouse. Sorry, my mouse isn't working. This is the RNA transcript being made. It's very small. That's the DNA. And this one, I don't even see any RNA transcripts being made yet. All right, any questions about transcription? So once again, the RNA polymerase, which transcribes the gene, binds to the promoter. As I stated, RNA transcription cannot happen on a double-stranded DNA. So you have to make the DNA single-stranded. And the RNA polymerase has an enzyme, meaning this is a complex of many different proteins, binds to the DNA. And the first protein up here actually unwinds the DNA because it has a helicase activity. So the first protein, you can call it a helicase. And the RNA polymerase is a combination of different proteins. We're not going to talk about any of the others, but there's like a stabilization factor and an enhancement factor, and obviously the helicase, as well as the RNA polymerase. The RNA polymerase then moves down the DNA, inserting RNA in the growing molecule of RNA. Let me see if I've got that in a little better format. Not really. I'll just try and blow it up here. So the RNA polymerase binds the promoter, which is right there, and then it moves down the DNA, adding RNA, which I don't see any there, but there the light green is here. And it moves down the DNA, inserting RNA. Initially, that RNA will hydrogen bind to its complementary DNA. So initially, the RNA is hydrogen binding to the DNA. Later on, though, the RNA starts pulling away and it will not be hydrogen bound to the DNA. But initially, the RNA is first made. That RNA is hydrogen binding to the DNA. And it's a single strand of DNA. The other strand is looping out. And usually, at least in eukaryote DNA and most prokaryote DNA, the gene is on one strand of DNA and only that strand of DNA will be transcribed. And a virus that differs. Both strands of DNA in a virus can code for information, in which case we would have to have both strands of DNA. We're talking about a double-stranded DNA virus, which we'll talk about that later too. But in a prokaryote as well as a eukaryote, usually only one strand is transcribed because usually only one strand has the genetic information. The other strand is just complementary to it. And then see if I can burrow that down. The RNA polymerase moves down the DNA, transcribing new RNA. And you can see here that the start, so maybe like 3 loops of the RNA is actually hydrogen binding to the DNA. But then we have a large number of loops, about 5 or 6 over here, where the RNA is not bound to the DNA. And what happens here is that the DNA snaps together so that the single strand of DNA comes down and then rebinds to the DNA. But the portion being transcribed would be this portion here, and it's only single-stranded. But this portion there, the DNA snaps back together and is double-stranded. The RNA polymerase proceeds down the gene all the way until it binds to the terminator. The terminator codes for the RNA polymerase to fall off the DNA. That will release the RNA, and then the DNA will snap back together and become double-stranded once again. Any questions about transcription? All right, let's watch a little video on transcription. Oh, shoot. Oh, there it is, I think. Let's see what this is. This is a lac operon. This might not be what I want.
The E. coli lac operon is an example of an inducible set of genes.
Yeah, this we'll keep it because we'll look at it later. Probably not today though. But let me look up transcription. I thought I had updated that and remade it, but that might be a little too short, but let's try it.
How to get the most of Grammarly free. It's Kayla on behalf of Grammarly to tell you egg proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. Motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand.
As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons.
Yep, let's stop that here. Well,
The genetic code includes
yeah.
64.
all right, we'll get to that. That's protein translation. This was not a very good model of transcription. It was going into a lot of I don't know, complicated information that I normally don't talk about, and I apologize. I've got to find a good video, and I just, like I said, this keeps changing. It's on YouTube, and I thought I just redid that last term though. So let me see if I just happen to have a link here that works. Yeah, that one's not there either. All right. Any questions about any of that? Anyways, I know not to get that one because it covers some material outside of RNA transcription and goes into a lot of complicated information too. I don't see the one that I used to use, but obviously it's been moved or taken down because it's not there anymore. Oh well, let's move on. Any questions about transcription? If not, let's move on to translation. Translation is the synthesis of protein by ribosomes using the information in the messenger RNA. Messenger RNA is translated in codons. A codon is a group of 3 nucleotides. Translation of messenger RNA begins at the start codon, and the start codon is AUG. And as far as I know, that's always the case, at least in humans and in eukaryotes. The start codon tells the ribosome, begin protein translation here. The start codon, or AUG, also codes for the addition of the amino acid methionine. So every protein that is first made, when it's first made from the RNA, messenger RNA, it will always start with the amino acid methionine. And if that protein doesn't want to start with the amino acid methionine, it'll have to modify the protein and remove that methionine. Any question about any of that? Translation will then proceed down the messenger RNA until the ribosome moves to one of the 3 stop codons. And you do not need to know the stop codons, you don't need to memorize them. You should just know that there are 3 different stop codons, UAA, UAG, and UGA. When the ribosome reaches a stop codon, it will stop adding proteins to the growing polypeptide chain that we'll call the growing protein. And then the ribosome will fall apart, releasing the growing polypeptide chain that we'll now call the finished protein. And protein translation, of course, will end. Any question about any of that? Now, obviously, that's in a summary. We'll go into it in more detail. No questions? I'm moving on. The information in messenger RNA is encoded in a codon, and the genetic code of the codons is shown here in this table, where each messenger RNA codon is a sequence of 3 RNA nucleotides, and these 3 RNA nucleotides code for 1 amino acid. Organisms, at least humans and most organisms, contain about, well, exactly 20 different amino acids. At least for humans, it's exactly, there's 20 different amino acids. But the number of codons would be a little over 60. Let's see what that is. 1, 2, 3, 4 times 4 is 16 times 4. Whatever 16 times 4 would be is how many number there are. Let me do that. I've always wondered, how many are there exactly? So 16 times 4. I shouldn't do this now because I'm trying to catch up, but I'm just curious. Oops, shoot. Didn't do that right. That's still not right. Hit that one there. So there's 64 possible codons. So let me just change that in the PowerPoint. So there are 64 possible codons. So obviously, if there's only 20 different amino acids, but 64 codons, something has to give. And what happens is that there's more than one codon coding for most of the amino acids. So more than one codon for each amino acid. There are, I should say, 2 exceptions. Let me blow this slide up so I can show it to you. Methionine is only coded by AUG. So wherever in a protein you want methionine, you got to put AUG in as the codon. And then there's one other exception. I think it's tryptophan. Yeah, tryptophan is UGG is the only codon coding for tryptophan. All of the other codons have 2, 4, or 6 codons coding for that amino acid. So phenylalanine, there are 2 codons coding for that one amino acid. For serine, there are 4 amino acids coding, 4 codons coding for that one amino acid. And then for leucine, there are 6 codons coding for that one amino acid. This this arrangement where there's more than one codon coding for some of the amino acid is known as the degeneracy of the genetic code. Any question about any of that? All right, let's watch the translation video.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Okay, any questions about that video? So let's go through protein translation. Remember, DNA is made into messenger RNA, and then the messenger RNA can be translated into protein. What happens is the messenger RNA moves to a ribosome, and that'll always be in the cytoplasm. And then the 2 ribosome subunits come together on the messenger RNA, and so they'll bind here. And then tRNA brings in the correct amino acid. It's actually tRNA that does the reading of the messenger RNA. And the correct tRNA for AUG would be the tRNA that has the anticodon UAC. So the U binds to the A binds to the U, and the C binds to the G. And that's why we say the tRNA is actually the one reading the ribosome. The ribosome is just holding everything together and then moving down the messenger RNA, moving a codon at a time. The ribosome, although we do say that it reads the messenger RNA and it's the one doing the protein translation, in reality, the ribosome does not read the messenger RNA. It's the tRNA that does, with 1 or 2 possible exceptions, and that is the ribosome will read the AUG, and it will read the terminator as well. Not the terminator, the stop codon, sorry, the stop codon. And it will not put on any amino acids upstream of AUG. So the ribosome will not begin protein translation until AUG, and then they'll bring in the correct tRNA to bring in the correct amino acid. So upstream of the AUG, the messenger RNA does not code for any protein. Any question about that? And conversely, downstream of the stop codon, the ribosome does not put on any amino acids. So downstream of the stop codon, the RNA of messenger RNA is not read. Any question about any of that? So what happens is that the ribosome assembles on the messenger RNA, and it has two channels in it. The first channel, you don't need to know the name of it. It does have a name. I think it's the P site or the A site, something like that. And the first tRNA comes in, and it has to have the anticodon UAC to bind to UAG. And the first tRNA brings in the correct amino acid. And that's why AUG codes for the amino acid methionine. And methionine is only on the tRNA with the anticodon UAC. Any questions about all that? Now, the ribosome is extremely important because obviously it's reading the AUG and beginning protein translation here. It's also moving down the messenger RNA one codon at a time. And it has this channel in there that stabilizes the tRNA. So this tRNA is stabilized by the ribosome. And I don't know if it's hydrogen bonds or ionic bonds. I don't think it's a chemically covalent bond, but the ribosome stabilizes the tRNA in this position, and that allows the tRNA to come in, and then the anticodon binds, hydrogen binds to the codon of messenger RNA. The second tRNA then comes in the second channel in the ribosome, and it does the same thing, stabilizing it and allowing it to come in. And the second channel, allowing the second tRNA to come in, has to be the tRNA with the anticodon that is the complementary to the second codon. And the second codon can be any of the codons coding for any of the different 20 amino acids. It could even be AUG. You could have AUG and AUG, again, in which case it would bring in methionine. But the point is this tRNA brings in the correct amino acid that this codon codes for. Any questions about that? Then the RNA portion of the ribosome removes the amino acid from the first tRNA and then binds that first amino acid with a chemically covalent bond to the amino acid of the second tRNA. And that starts growing the polypeptide chain. When it's all finished, we'll call it the finished protein product. Once again, that RNA portion of the ribosome is a ribozyme, an RNA enzyme that does this. And that's the only RNA enzyme we'll talk about in this class. I think your textbook doesn't mention that either. I'm just mentioning it. All right, so this first tRNA now does not bind to an amino acid. and that'll allow the tRNA to fall out of the ribosome, come out, and that'll open up the first channel. And as I stated, the first and the second amino acids are now bound. This will then allow the ribosome to move down to the next codon. So when it moves down, the first channel region of the ribosome will now be over UUA. And so this will be moved to the first channel. And there we see it there, where this is now moved to the first channel. That will allow the third amino acid to come in, and it brings the correct amino acid, which in this case is glycine, which is what GGU codes for. And how that happens is glycine is on the tRNA with the anticodon CCA. Once again, the ribosome enzyme of RNA, meaning the ribozyme enzyme, will remove the amino acid leucine from its tRNA and then bind leucine to the newly added amino acid. That will mean that this tRNA has no amino acid bound to it, and so it will fall out. And as it falls out, that will tell the ribosome to move down one codon. And once again, what was in the second position moves to the first position. That allows the second position to bring in the correct amino acid. In this case, it'll be the tRNA carrying the amino acid methionine, because AUG codes for methionine. And yes, you can have AUG more than just the start position. Wherever you want the amino acid methionine, you have to have the amino acid AUG. Usually, only the first AUG codes for starting protein translation here. And I'm not going to talk about the exceptions. So anyways, we now have a growing polypeptide chain, and this will continue to grow as the ribosome moves down one codon at a time, adding in another amino acid. This will continue this way until the ribosome reaches a stop codon. The stop codon codes for the ribosome to stop protein translation here. The ribosome will fall apart. That will release the protein from the tRNA and the ribosome. And then the ribosome itself will fall apart, and the messenger RNA will be free. And that will allow another ribosome, or maybe the same one, to bind up here once again to the messenger RNA and then translate it again. This protein, when it first comes off, all it is a sequence of amino acids. But shortly after it falls off, the nearby amino acids will start interacting with each other, causing the secondary level of the protein to form. And then once that's finished, the amino acids all over the protein will start interacting with each other, causing new reactions, and that'll be the tertiary structure of the protein. Any question about any of that? All right. So once again, you can call that the growing protein. You cannot call it the protein until it's finished. And then the final polypeptide is the final protein, and that's what we call a protein. Any questions about any of that? All right, here we're seeing protein translation occur in the cell. This is an actual electron microscopic image. So there's the DNA. Here's the artist's rendition. This is the DNA. And you can note that in cells, we frequently will have more than one ribosome bound to the same messenger RNA. And that means that this will be making a protein, 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 that will be making a protein. Any question about any of that? Because that's the ribosome, and this has 3 ribosomes on it, and they're each beginning to translate the messenger RNA. In a prokaryote cell or a bacteria, we have some complications, and that is the DNA and the RNA and the protein translation are all happening in the cytoplasm of the cell. And so this one messenger RNA, well, let me see about that. That's the DNA, sorry. This messenger RNA here can have more than one ribosome bound to it, and each ribosome will build one protein. And so this one will have currently 2 growing polypeptide chains, which will make 2 proteins. But as you can see, we can have more than 2. Like this one has 1, 2, 3, 4, 5 different ribosomes bound to it. And in bacteria, we have a further complication, and that is the DNA is in the cytoplasm, and transcription happens in the cytoplasm, as well as translation happening in the cytoplasm. So here we can see this DNA has 1, 2, 3, 4 different RNA polymerases making 4 different RNA molecules, messenger RNAs in this case. And what I'm trying to say is that before RNA transcription ends, we can have protein translation beginning. And that's because everything is happening in the cytoplasm. In this case here, we have, well, that one there, 1, 2, 3, 4, 5, 6, 7, 8 different RNA polymerases making 8 different messenger RNAs. And each of them has more than one ribosome bound to the RNA. So we're having, in this case, 1, 2, 3, 4, 5, 6, 7, 8 different messenger RNAs being made on that one gene at the same time, and countless numbers of protein made, because this one has like 3 proteins being made, that one 1, 2, 3, 4, 5, 6 proteins being made, and this one I don't even want to try and count it. A large number of proteins being made, because each of these is a ribosome bound to the messenger RNA, translating that messenger RNA into protein. The important part is that you can have more than one polypeptide chain being made from the same messenger RNA, and then you can have protein translation beginning before RNA transcription has ended. And so you can have multiple RNA transcript units, as well as you can have multiple growing polypeptide chains for each messenger RNA. This is a complication only seen in prokaryotes, specifically in bacteria, because that's what we're talking about. You don't see this complication in eukaryotes because you cannot begin protein translation before RNA transcription ends in a eukaryote. And that's because RNA transcription happens in the nucleus. That RNA then has to move into the cytoplasm before protein translation can occur. However, you can have in the nucleus more than one RNA molecule made at the same time from the same gene. So that complication can happen. And then once that messenger RNA moves into the cytoplasm, you can have more than one ribosome bind to it. So you can have more than one growing protein or growing polypeptide chain forming on that one messenger RNA. Any question about any of that? Sorry about the complication, but there are actually several happening in a bacteria, and there's a few happening in a eukaryote. Eukaryotes have another complication, and that is they have their genes, which are transcribed, composed of exons and intron, regions of the gene. An exon is a region of the gene that actually codes for the final gene product, meaning for a enzyme or a protein, the exon actually codes for the amino acids of the protein. The introns do not. They do not code for anything about the protein. And when in a eukaryote the RNA is transcribed, it has both exons and introns in it. This RNA molecule cannot leave the nucleus, meaning the nuclear pores, meaning the what the messenger RNA moves through to get into the cytoplasm, the nuclear pore, has a check, and any messenger RNA which has introns are not allowed through the nuclear pore. So the initial RNA transcript stays in the nucleus. And it's not until the introns are removed and the exons brought back together that this finished messenger RNA can now leave the nucleus, meaning the gateway pore notices that it has no introns and it will allow it to leave the nucleus. How that happens is the introns are actually cut out. We have a term for that. It's called splicing. So you splice here and you splice there, and then you splice together this exon to that exon. And that means link them together. So splicing is both used to cut the introns out, and then splice is also used to bring the exons back together. And by bringing them back together, we're saying combine them in a chemical covalent bond. Anyways, that's a complication in eukaryotes where they have both introns and exons within the gene. All right, so we've talked now about the structure and function of genetic material, the flow of genetic information, DNA replication, RNA transcription, and protein translation. It's now time to talk about the regulation of bacterial gene expression. We're going to use the operon model to discuss the regulation of bacterial gene expression. Bacterial gene expression is similar to eukaryote gene expression. It's just a little simpler, and there's less regulation of bacterial gene expression than of eukaryote gene expression. Any question about any of that? All right. So the cell wants to regulate its contents, and one way to regulate its contents is to control gene expression. We have some enzymes, constitutive enzymes, which are always needed by the cell, and they're expressed at a fixed rate, meaning the genes are always turned on. There are other enzymes which are expressed only as needed, and these genes are turned on when they are needed and turned off when they are not. and those we call repressible or inducible enzymes. And we'll talk a little bit more about what's the difference. You can regulate the contents of a cell in other ways, like there are pre-transcriptional control, and we'll talk about inducible operons, but there are also repressible operons, and we're not going to talk about that, and you won't be tested on it. There's also epigenetic control mechanisms that control gene expression, and it's really hairy. And even today, it is not well understood. And people have been trying to understand gene expression for at least 50 years. Let me see now. I was in undergraduate school in the 1980s. And they had been looking for trying to understand epigenetic control at least for 10 years, maybe 20 years before that. And they didn't understand it then, and they still don't understand it, at least fully today. They do have some understanding, but not full understanding of epigenetic control. And then there's post-transcriptional control means too. We won't cover that, and you won't be tested on it. And how you can do post-transcriptional control is you can make the enzyme, and then you can shut down the enzyme, make it non-functional. And one way to do that is you can simply have a inducible repressor bind to it. Any questions about any of that? All right. Getting back to repressible and inducible enzymes and constitutive enzymes or genes. Like I said, some genes are always turned on, and that's because their gene products are always needed by the cell. Other genes are only turned on when they're needed, and they're turned off when they're not needed. Because if the cell doesn't need the gene product, why spend energy making it? It doesn't make sense to make a gene product if you're not going to use it. All right, any questions about any of that? If not, I think I'll stop here and we'll continue with this next Tuesday.
Thank you for your time.
All right,
Have a good night.
you too.
Thank you.
You're welcome.